there are still patients who progress and relapse multiple times despite all these therapies, and we're left looking for new options that are coming. I'd just like to spend the last few minutes talking about what's out there that's got you excited about what's coming, Bill, or have we seen the...
Well, one drug that I think is quite interesting are the checkpoint inhibitors, PD-1. And while the initial therapy with monotherapy were pretty disappointing, there weren't any real partial responses among a study with one of the checkpoint inhibitors. There was a data presented at ASH combining it with lenalidomide and dexamethasone, and they saw very impressive response rates, above 50%, even among patients who were known to be refractory to lenalidomide. So the idea that you could recapture sensitivity by adding in a PD-1 inhibitor, to me, is very intriguing.
I think there's an update at this meeting on that too,
I think
right?
there will be, but I think that's very exciting.
What are the other things? Do you think checkpoint inhibitors are going to work in myeloma? They work in Hodgkin's disease, they work in a lot of solid tumors.
So, I mean, I think they will. I mean, I think the principle here is that immunotherapy is not disease-specific or tumor-specific. And so I think we've seen globally that immunotherapeutics is going to be important in all cancers. Now, how do you use immunotherapeutics is going to be the question. So things like prostate CA, which are not immunogenic, and we saw no responses with a single agent with a PD-1 inhibitor, when you combine it with a CTLA inhibitor, there's actually some very nice robust activity that we see there. So I think similarly in myeloma, it's not the most immunogenic tumor like melanoma that we can expect to see single agent activity with a checkpoint inhibitor or a PD-1 inhibitor. But I think clearly as we move forward, now there's some very intriguing data with IMiDs, and we have to generate the data with proteasome inhibitors. We have to combine it with other checkpoint inhibitors. And so we're going to be doing a trial at MD Anderson now where, similar to melanoma, where they combined a PD-1 plus a CTLA, we're going to be doing that in myeloma as well. So I think that that's very exciting as we move forward now, that immunotherapy globally is going to be an important part of myeloma.
Yeah. I guess I'm a little bit more on the fence about checkpoint inhibitors personally, but what do you think, guys?
You know, I think when you look at checkpoints, these are the only other agents that are going to give us a flat curve if what we're seeing in other tumor types holds. The fact that it rescues lens sensitivity, I think, is interesting. Almost all the agents we have talked about before, they have had a duration, even if that's in years. So I'm at fault for being too optimistic, but I do think the checkpoint inhibitors do hold a lot of promise for myeloma.
Well, you've got some trials at your center.
Well, we have interest in immunology across different diseases at our institution, and I think there is a lot of preclinical data that would strongly encourage doing it, but then at the same time, there is no single drug activity. So combining it with which drugs? Should it be with IMiD? Should it be with daratumumab? Should it be with other types of cell-based therapies? Could it be with CAR T-cell therapy?
Yeah, talk about them for a minute, because I must admit I'm on the phone now with NIH in Pennsylvania trying to find slots for CAR T-cell therapy. What do you think of those treatments?
So we are opening our first trial. We're going to put the first patient on the CAR T-cell for BCMA March 1st of 2017. That's when we are going to open our CAR T-cell program for myeloma.
Next year.
This is going to be next year. So we are very enthusiastic about it. We have followed closely the other groups, and our institution has a track record in acute leukemias and other diseases as well. So myeloma will be our next disease where we're going after with CAR T-cells. And we intend to do armored CAR T-cells where we're going to clone in interleukins into the T-cells. And we also are planning on doing more than one target in the CAR T-cells. So you don't only have BCMA, you have 2 or 3 other targets as well.
You're getting pretty fancy.
Wow.
Okay, what other drugs are out there? Rafael, what else has caught your attention? What about venetoclax or selinexor?
You know, I'm particularly keen on venetoclax. I'd like to see a lot more work being done.
Maybe I jumped right into it. Tell us what that drug is.
Yeah, so it's a new compound, which BCL2 inhibitor, but it's interesting because we have seen on a preliminary fashion that patients with a translocation 1114 or with lymphoid morphology seem to respond very well. I'm actually eager to start using it on some of my patients that have exhausted all other options. Their response rates have been very high, including complete responses. There's several trials going on, so I think it's being played out. I'm not sure what the best use or in what combination is best used, but I'm going to venture out to say that, particularly for patients with a lymphoid variant myeloma 1114, this seems to be a very promising agent.
Anybody else had experience with venetoclax?
I have not, no.
Yeah, I must admit, I guess we've had a trial, and I've had great deal of success with it, but mainly in the 11-14 patients, as you pointed out. So a real targeted therapy, approved for CLL now, but I think worth trying in your myeloma patients off-label. What about other, the selinexor maybe is one we should mention and talk about?
So I mean, I think continuing with actually the CLL drugs,
Okay.
just briefly, I mean, we see ibrutinib, which is also approved in CLL.
Do you think that's got activity in myeloma? Or?
Seen as a single agent, we didn't see much activity, and biologically, we're not sure why it would. But the clinical data from Ajay that was presented at ASH was kind of compelling, where we see some nice response rates with carfilzomib. So I think that, you know, I think that's some interesting data there that we need to follow as well. You know, the selinexor data also is very interesting as well.
What is selinexor, Jatin?
Yeah, so selinexor is a SINE protein inhibitor. So basically,
It blocks exportin.
exactly,
Yeah.
exactly. So I think that's a very different mechanism of action that we see with selinexor. But the challenge is going to be trying to find the right dose and the right schedule and the right combination, because it has a fair number of side effects that we see from fatigue and asthenia and GI toxicity as well.
Okay. Any other thoughts on selinexor? Some promise. It's got some single agent activity, it looks like.
I think it's a promising agent, like Jatin said. We are opening a trial now in the relapse setting together with ixazomib, where we are decreasing the dose of selinexor in combination with ixazomib, and it's too early, so I don't really have any data to share today.
Any other drugs out there that we haven't talked about that people would like to mention? Pipledepsin got some coverage yesterday. Anything catch your fancy there?
I have no experience with that drug.
I think the other exciting target as well is BCMA. I mean, I think that there's not too much clinical data quite yet, but that's an important other target on the surface of plasma cells that for our community physicians to be aware of, that we're seeing some trials now as a naked antibody, as a drug conjugate, as a BiTE technology targeting BCMA. You're using CAR T cells to target BCMA. So I think that's an important target. The way we attack it, either by naked or antibody, I think is going to be explored in the next few years. But I think BCMA as a target is an important one to keep in mind for our community physicians. I think that's high promise.